Welcome! Today I'd like to review for you my work in the lab of Dr. Gary S. and Jones on the effects of chronic cocaine self-administration on orexin signaling in the medial prefrontal cortex, or MPFC for short. The orexin system is characterized by the orexin neuron cell bodies exclusive to the hypothalamus and their respective neural projections. The Gary S. and Jones lab and other groups have established that the orexin plays a major role in addiction processes and may be a key therapeutic target. For my project, I'm interested in the medial prefrontal cortex, which is thought to regulate the anticipatory phase of addiction. My work is focused on investigating the changes in the orexin A peptide and the orexin receptor mRNA in the MPFC after cocaine exposure. By understanding the changes in the orexin in the MPFC, we could gain a better understanding of the relapse vulnerabilities associated with the orexin system. So, two separate cohorts of rats underwent jugular vein catheterization for chronic cocaine self-administration. Our first cohort of Sprague-Dawley rats underwent behavioral economics, which is a paradigm used to characterize addiction-like markers such as alpha and Q0. The Sprague-Dawley rats also underwent intermittent access, which is a clinically relevant drug use model that mimics human drug use. Our second cohort of rats, Long Evans rats, only underwent the behavioral economics portion. After these studies were conducted, brain tissue was harvested, cryosectioned, immunohistochemistry was done to quantify the orexin A peptide expression, and then in situ hybridization was done to quantify the orexin receptor 1 mRNA expression. Then, ImageJ, an image processing program, was used to analyze and quantify the positive detection from the captured images. So a big part of my project during my time with LSAMP was troubleshooting molecular instruments in situ hybridization platform. This technology utilizes a probe and hairpin system to detect desired mRNA sequences. When first using this platform, we had a lot of undesired nonspecific detection, and I was able to show how tissue autofluorescence, a nonspecific fluorophore amplification, is observed to be qualitatively different to a section with both probes and hairpins. Overall, I figured out that the processing conditions were paramount, and essentially, the fixed tissue must be humid at all times of the hybridization process. By utilizing the in situ hybridization, we found that there is a significant positive correlation between alpha and orexin receptor 1 mRNA in both the prelimbic and cingulate cortex 1 regions of the MPFC. This alpha value is demand elasticity, which is the inverse of demand motivation to respond to drug. And we get this value by knowing how much the rodent is willing to press to self-administer cocaine, while cocaine availability is decreased in a quarter logarithmic manner. We also found that there is a significant negative correlation between Q0 and orexin receptor 1 mRNA expression in PL and CG1. Q0 is known as a hedonic set point, which is the baseline preferred drug amount. These findings suggest that postsynaptic cells compensate for excess orexin peptide expression by decreasing the receptor availability. And, as we can see, compared to naive animals, intermittent access animals had increased orexin peptide expression in the MPFC on average. This suggests that intermittent access cocaine exposure either generates orexin fibers or already existing fibers happen to express more of the orexin peptides. So, we know changes in orexin signaling after chronic drug exposure may promote relapse vulnerabilities, and changes in the medial prefrontal cortex can alter higher cognitive function. So, by understanding how the orexin impacts the medial prefrontal cortex, we can start to unravel the mechanisms underlying motivational control of higher order cognitive capacities during addiction. For my future studies, I want to do immunohistochemistry to quantify the orexin receptor 1 protein expression. More studies are needed to dissociate between the ventral and dorsal regions of the MPFC during orexin signaling, and chemogenetic studies can be implemented to modify the orexin circuit to determine the orexin plasticity in the MPFC during cocaine self-administration. I would like to thank the Gary S. and Jones Lab, Dr. Jorge Avila, and LSAMP for their hard work, support, and assistance throughout this whole project. Thanks for listening.